Okay, so thank you again for the opportunity to speak here today, to introduce you to Calimmune and some of the clinical programs we have going on, and then again, opportunities for partnering. So Calimmune is a clinical stage company really focused on gene-modified cell therapy for hematologic diseases. We are not a new company, not a startup. We have about a decade of experience in this area. Our cell processing teams have made autologous hematopoietic stem cell products for over 100 trial participants. What I'm going to do today here is really highlight a couple of areas, one in manufacturing of lentiviral vectors that we call Siteegra T, and the other that is called Select Plus, which really addresses delivery and logistics. It's a mechanism to select and enhance engraftment of gene-modified stem cells. Calimmune really began as an HIV gene therapy company and was founded with that premise. But capitalizing on the experience there and our background in hematopoietic cell manipulation and processing, we're now expanding and ready to announce a number of new programs in hematologic disease. So really, what we're set up to do is change the paradigm of stem cell gene therapy and go from a very costly inpatient process with risky high-dose chemotherapy and a very complicated adverse event profile to something that can be administered in an outpatient setting. Low-dose conditioning, a way to pharmacologically control and monitor the conditioning that's necessary to achieve engraftment of stem cells. A limited adverse event profile, and our intent is to really try to do away with some of the severest toxicities associated with conditioning for hematopoietic stem cell transplantation, and these are usually issues around sterility and fertility, in a way to modulate cell delivery and to achieve your therapeutic window. So first, we got into the field developing approaches to treat HIV disease, and our approach that I'll get to a little bit later, but it was a stem cell, hematopoietic stem cell and T cell approach that involved genetic manipulation with lentiviral vectors. Since the indication is so large, HIV disease, our patient populations were in the hundreds of thousands, which is much, much higher than other indications that are being targeted by gene therapy using lentiviral vectors, easily orders of magnitude higher. So very early on, we realized the need for scalability and a look towards future commercialization of our approaches. So we developed and licensed in a stable cell line system for lentiviral vector manufacturing that we call Siteegra T. And we have been developing this for a number of years and are now outsourcing this for GMP manufacturing and developing a number of partnerships with other commercial programs to further expand upon the use of this, and really with the intent of developing a standard for the field. The benefits of a stable cell line production system are not just scalability and cost, but really a standardization of the process and a reproducibility of the process. And you go from a very inconsistent process to a standardized GMP compliant process. There are cost savings, of course, and safety savings, but the real benefit is the standardization and the lack of variability in the outcomes of the process. So just a little bit of data here and stuff that we've done to kind of kick the tires on this technology. This system is the only stable cell line system that's been used to produce lentiviral vectors for clinical trials. Vectors have been made using the Siteegra T system for 2 different trials in the ex-SCID population. And this is just some of our work with our clinical vectors, comparing a clinical lot of virus made by standard transient transfection. This is an optimized process that we use for GMP manufacturing, to lentiviral vectors made using this stable cell line system. And if you spend any time in this field, you know it kind of depends on how you ask the question and in terms of the answers you get. So there's lots of different ways that we can assess comparability between the two systems. And this is a small subset of the data that we had to prepare to convince the FDA that the products made with a stable system were similar to that used by more the transient, but also the kind of the gold standard in the field right now. So this is one way that we compared, and this is just looking at titer based on expression of a gene product on the surface of a cell. So it's a FACS assay. And you can see that the titer of the two processes is very similar. Two other ways we can do it, again, just looking at titer of vector preparations, but also potency of the virus. We can do PCR for gene transfer efficiency, a measure we call VCN, or vector copy number. And then also activity assessments. And in our clinical program, in this instance, we are delivering a short hairpin RNA which knocks down CCR5 expression, CCR5 being a co-receptor for HIV. So this is our HIV drug product. But you can see in this instance that by both of these measures, we actually get a more potent vector made using the Siteegra T system than that made by standard transient transfection systems. And then for those that are interested in more of the scale up and bioproduction things, one of the problems that has really hampered the development of other stable cell line systems for viral vector production has been their lack of stability and reproducibility. So this is two different cell lines that I show over on the left there in the blue bars, two different cell lines that have been carried in continuous passage here for months, in excess of 3 months. We've actually carried these studies out to 6 months, but this is about 3 months of data looking for the stability of viral vector production, and you can see that it's stable over this time. We've also adapted these for production and growth without serum, suspension, adherent. These are all capabilities. And in the bottom right there, this is a typical bioreactor run at a moderate scale. This is actually the scale that was used to produce clinical vector for the trials that I mentioned. And you can see that it's an inducible system, and we get vector production this way in a scalable sort of system. So this is really one of the platform technologies that we took on to address the critical need that we saw in terms of scalability to hit our target patient population in HIV. But it's something that we're very interested too in partnering and have a number of programs along that line. Because of our experience and history with hematopoietic stem cell manipulation, isolation manipulation, growth, culture, we have unique capabilities in this area just through that wealth of experience. We do our own internal cell processing. We have a number of cell processing facilities and teams that do this, and have been quite efficient in terms of transduction efficiencies that we get, the potency of our vectors, stem cell collections, and that sort of stuff. So this kind of just illustrates some of those capabilities of Calimmune. The other new system that we've developed is what we call Select Plus. And this is really an attempt to change that paradigm in hematopoietic stem cell transplantation from an inpatient to an outpatient setting. Our Calone, our clinical program in HIV, we've actually done it all outpatient, in that we've done reduced intensity conditioning on an outpatient basis, and it's the first time that's done. But this is an attempt to move that a little bit further and to have a little bit more control over the process and to achieve higher levels of engraftment with reduced intensity conditioning. So Select Plus system is in a way that we can select positively for transduced cells. And we can do this both ex vivo and in vivo to drive the engraftment of gene-modified cells. So again, the intent is to change a standard paradigm from an inpatient hospitalization to an outpatient sort of process. It improves options for scalability and, of course, adverse event profile associated with toxic conditioning regimens. So show a little bit of data here. This is just a mouse study, again, looking at engraftment of human cells in mice. And you can see that there's both a positive selection for gene-modified cells with the Select Plus system and a negative selection against unmodified cells. So by putting this together, we have about a 2-order of magnitude window that we can play with based on dosing of the drug, which selects for these things. So that's a great opportunity that we're in the process of translating into the clinic. The analogous here is that we also have a select minus, a way of negatively selecting. It uses the same mechanism of the Select Plus, but in this case, you select against the gene-modified cells. So in this example, it would be analogous to a safety switch, which are being incorporated in a number of viral vector systems for a number of different indications. So this is both a positive and a negative selection capability, again, just depending on the pharmacological agent that's used to drive selection. And here, data in cell lines in primary mononuclear cells and hematopoietic stem cells, showing that you can not only select for these cells, but you can select against them. So again, as I said, Calimmune began as an HIV gene therapy company, and this is our most advanced clinical program. We have 3 ongoing clinical trials in our Cal1 program right now. It's an ex vivo autologous cell therapy using a self-inactivating lentiviral vector, which I already introduced you to. The mechanism of action is a short hairpin RNA against CCR5, co-receptor for HIV. It also expresses a protein that we call C46, which is a membrane-anchored fusion inhibitor for HIV. In preclinical models, it's been shown to be active and to protect against all tropic forms of HIV, including drug-resistance forms. And the intent here is really to model that genetic situation that occurs in a very small percent of the population, which is absence of CCR5. And it's been shown that folks that are homogeneously deficient for CCR5 are resistant to HIV infection, at least with the initial infection with an R5 tropic virus. So this is an intent to bring that to a wider population. And our initial trials are in a large unmet need segment, and that's art therapy interrupters. These are folks that, for one reason or another, have chosen to come off their antiretroviral therapy. And it's a very large market, again, greater than 100,000 folks just in the U.S. That first trial is in the final cohort right now. It's just gone to follow up in the U.S. And the preliminary data we expect within a few months here next year. So this is really, the trial was really predicated on some rather, exciting preclinical studies, and I'll just show a few of those. This is a non-human primate study. It's originally set up as a safety study to look at the engraftment of our Cal1 modified cells in a non-human primate model. So they were, stem cells were collected, they were manipulated ex vivo with our lentiviral vector, transplanted back in, and we had stable engraftment of gene-modified cells. We then asked the question, what happens if we challenge these animals with HIV? I can say we used a very pathogenic strain of virus. If control animals were infected, they would all progress to AIDS and need to be sacrificed, usually very short period of time. Certainly within 3 months, all these animals are dead. What you can see here, looking at T-cell counts following the viral infection, which is at time 0, they immediately plummet. So again, it's a very pathogenic strain of HIV, and they go down to a quite low level. But then they immediately start coming back up. So by protecting cells from the virus, we're actually encouraging selection for gene-modified cells. And you can see that the T-cell counts rise to within a normal range, which is shown by the dashed lines, and they've maintained at that level now for well over a year, close to 2 years in these animals. Similar, if we look at viral load, and this was very exciting to us. During the acute infection, these animals are clearly infected. We have extremely high viral loads in the plasma of these animals. But rather quickly, it goes down, and now we're out to 2 years, 2+ years for these animals. And there's one animal that more often than not, we're unable to detect any virus at all in the plasma of this animal. So just from protecting cells from viral infection, we're able to drive the virus down to a level of extinction, which is a great, kind of vote of confidence for the approach, but it's really unprecedented and something that we hadn't really expected. And it now, of course, is the subject of ongoing clinical programs. And so this is our Calone trial within the ART interrupter cohort. It's 12 patients, 3 cohorts looking at dose escalation for conditioning, but always with a reduced intensity conditioning and always in an outpatient sort of setting. Centralized cell processing facility that we manage and run in the United States here. We are dosing our last patient in this trial this week, actually. So what we've learned so far, really around the safety and feasibility of the approach. We've met all the parameters for cell processing and safety assessments. This is a combination product. Not only are we modifying hematopoietic stem cells, but T cells. That was a first. There are 2 gene products in the vector. That was a first. And really just, excuse me, the ability to conduct outpatient conditioning in non-malignant patient population was a great achievement here. We spent a lot of time in a very iterative process in optimizing our cell manufacturing through this trial and have achieved, of course, promising biologic activity, which we hope to report on in the coming months. So just in summary here, Calimmune has been around for about a decade. We have a very experienced team in the cell and gene therapy space. Novel classes of therapeutics, really capitalizing on one-time treatments in hematologic diseases, ex vivo autologous cells primarily. We have the Select Plus technology to drive selection and engraftment of gene-modified cells, coupled with the Siteegra T platform as a proprietary lentiviral vector manufacturing system. Strong clinical pipeline, really proof of concept in HIV/AIDS, and now expanding to candidates in other ex vivo gene therapy sort of situations, settings. A number of academic, government, and private partnerships that have helped propel this program along. And with that, I'll finish. Thank you.